ever looked in the mirror and seen, I don't know, your mom's eyes staring back at you, but with your dad's curly hair? It's a classic family puzzle, right? How do we end up with this totally unique mix of traits? Well, today we're going to crack that exact code by diving into a brilliant experiment from the 1800s that completely changed how we think about genetics, the dihybrid cross. So here's our game plan for this explainer. First, we'll dig into the big question that had early scientists totally stumped. Then we'll define our main tool, the dihybrid cross, and see it in action with Gregor Mendel's now famous pea plants. We're going to get to the bottom of that mysterious 9:3 to 3:1 ratio he discovered, and then we'll see why this idea from over 150 years ago is still a massive deal in science today. All right, let's just hop in a time machine for a second. Before we had any concept of genetics, heredity was, well, it was a complete black box. It really seemed like a lottery. You got some traits from one parent, some from the other, but the rules behind it all, a total mystery. And that led to the million-dollar question, are traits inherited as a package deal? Like, if a pea is yellow and round, does that mean its babies will always be yellow and round? Or can nature kind of shuffle the deck and create brand new combinations? That is the exact puzzle we're here to solve. And enter our hero, Gregor Mendel. In the mid-1800s, this Austrian monk, just quietly working away in his monastery garden, was about to uncover the fundamental rules of life itself. His simple, elegant experiments with pea plants would lay the foundation for, well, all of modern genetics. No big deal, right? So how did he do it? How did he figure all this out? He designed this beautifully simple yet incredibly powerful experiment. And that brings us to the star of our show today, the dihybrid cross. Okay, so a dihybrid cross is really just an experiment that tracks two different traits at the exact same time. The di in the name literally means two. But the real key here is that the parents are heterozygous for both traits. Now that's just a fancy science word for saying they carry two different versions or alleles for each gene we're looking at. So one for round seeds and one for wrinkled seeds, for example. And to really get why the dihybrid cross is so cool, it helps to compare it to its simpler cousin, the monohybrid cross. See, a monohybrid cross only looks at one trait, like flower color, and it gives you this simple 3:1 ratio of dominant to recessive. But by adding that second trait, the dihybrid cross just unlocks a much richer story. It reveals a way more complex 9:3 to 3:1 ratio, and a completely different law of genetics. So what did Mendel's experiment actually look like in the garden? Let's get our hands dirty, metaphorically speaking, and walk through it step by step, because this is where the magic really happens. Okay, so Mendel picked two really simple traits, seed shape, either round or wrinkled, and seed color, yellow or green. He started out with what we'd call purebred parents, one that always made round and yellow seeds, and another that always made wrinkled and green seeds. When he crossed them, all the kids, that's the F1 generation, were round and yellow. But here's the brilliant part. He then crossed these F1 plants with each other to see what would pop up in the next generation. Now, to predict what's going to happen, we first need to figure out what kind of reproductive cells, or gametes, those F1 plants can even make. Think of it like this. Each parent has a grab bag of alleles for shape, big R for round, little r for wrinkled, and another bag for color, big Y for yellow, little y for green. When it makes a gamete, it pulls one allele from each bag. So you could get round and yellow, round and green, wrinkled and yellow, or wrinkled and green, 4 possible combinations. And this is how we map out all the possibilities. We use a tool called a Punnett square. We just list the 4 possible gametes from one parent across the top and the 4 from the other parent down the side. Then you just fill in the grid combining them. I know it looks a little complicated, but it's really just a simple grid to see all 16 potential genetic combos for the grandkids, the F2 generation. It's like a prediction machine. This, right here, is the moment of truth. After meticulously counting thousands and thousands of pea seeds, Mendel found something extraordinary. It wasn't random chaos at all. There was a hidden pattern, a beautiful, consistent mathematical ratio. He found this exact ratio, 9:3:3:1. This wasn't a fluke. This wasn't a coincidence. This was a fundamental law of nature whispering its secrets right from his garden. So, what on earth does that mean? Well, it means that out of every 16 offspring, on average, 9 were round and yellow, 3 were round and green, 3 were wrinkled and yellow, and just 1 lonely pea was wrinkled and green. But here's the kicker. Notice those new combinations, round and green and wrinkled and yellow. Those combos didn't even exist in the original purebred grandparents. The appearance of those brand new combinations, that was the proof Mendel was looking for. It meant the genes for seed shape and seed color weren't linked together. Nature was shuffling the deck. This became Mendel's law of independent assortment. Basically, the alleles for one gene get sorted into gametes completely independently of the alleles for another gene. Okay, so we've seen how Mendel cracked the code with his humble pea plants. It's a great story. But why does this 150-year-old experiment still matter so much today? Let's bring it into the 21st century. You know, this isn't just some old theory in a textbook. It's the absolute foundation of modern biology. In agriculture, scientists use these very principles to breed crops that are, say, both high-yielding and resistant to disease. In medicine, it helps genetic counselors predict the chances of a child inheriting certain genetic conditions. It's the starting point for understanding the incredible diversity of all life on Earth. Mendel's law of independent assortment is a cornerstone of genetics, for sure. But, you know, like all good rules, there are exceptions. And that leaves us with a really tantalizing question. What happens if two genes are located so close together on the same chromosome that they do travel as a package deal? How does that change the whole game of inheritance? Well, that's a story for another time.